Here's a picture of the tip of an onion root. So it's right at the end of onion root, and because it's a growing root, it's going to have a lot of mitosis taking place for growth. Some of the cells will be in interphase between divisions, and some of the cells are going to be in mitosis. So let's see if we can identify some in mitosis. We can see over here, this cell here, possibly here, and so we can go through and identify all the cells where it's obvious that mitosis is taking place. Some of the other ones it's harder to tell, but this gives us a sense of how active the tissue is in terms of its division. Now we can see that most of the other cells here are not in mitosis. And so if we count these up, the ones that are definitely in mitosis, and it doesn't matter which stage they're in, we could go through and try and identify some of the stages. This one here, they're being pulled away, so this is anaphase. Over here they seem to be condensing, so this might be prophase. Anaphase, 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 anaphase. Over here they seem to have been pulled apart, so that might be telophase. They're lining up here, metaphase, metaphase, anaphase there. Here they seem to just be condensing, so maybe prophase, and these are pulled apart, that's telophase. So if we add them all up, we get to a number. We get 13. Now if we do that number, which are in mitosis, the number of cells which are in mitosis, out of the total number for the whole picture, whatever that might be, then that will give us our mitotic index. So a mitotic index is the number in mitosis divided by the total number. Now let's say that number was 100, we would do 13 over 100, and that would give us our mitotic index, which is going to be 0.13. So the higher that number, the more active the tissue is in mitosis in terms of growing, dividing. Here's our summary. So the mitotic index is all the cells in mitosis, any of those stages, prophase, metaphase, anaphase, telophase in cytokinesis, divided by the total number of cells. And that gives us an indication of how many cells are in a state of cell division. And we get a high mitotic index in root tips, shoot tips, and other regions of cell division and growth.